squids and octopi, bivalves. I really can't get into Metazoans. I once wrote a paper about them. But the Metazoans, as you know, are a diverse group. And I have an outline, and it gives you the salient features, and it kind of gives you a little classification guide. As the protozoans, I don't expect you to leave here knowing everything about worms. But I expect you to have some kind of basic knowledge so you can then use the document that I gave you to try to identify the worms yourself. You know, for years when people told me, well, the worm has a large intestine. Large is such a relative word, if you don't know what large and small is, you can't tell that they have, you know, what kind of intestines. So today, that's what we're going to go through. We're going to go through the groups. We're going to start with the nematodes because I like them the best, and I'm talking. We're going to go through the nematodes first. All right? I'm going to first go through basic characteristics, large intestine, small intestine, Platy-Myerian muscles, Celomirin muscles, all of these things. And then I'm going to go through the groups. The information that I'm going to talk about is all in your handout, but if you're wanting to take notes, go right ahead. And then we have some slides at the end about new frontiers in veterinary pathology. Okay, people, these are nematodes. Now, we're going to move through this stuff because I have a lot of slides. Nematodes usually have a body cavity which is called a pseudocoelom, which you really can't tell from a coelom, but since it is a nematode, it's called a pseudocoelom. But it's a space, huh? And it has things in it. But sometimes the musculature and the lateral cards fill everything, and you really can't appreciate that space. I'd like to first start talking about the cuticle. When you describe a worm to your contributor, start on the outside and go to the inside, hitting the salient features. Okay? That's the way good parasitologists do it. Now, cuticles can be, this is a cuticle right here. This is thick. All right? Do you see a cuticle here? No. It's so inapparent, it's thin. So that's thick. Thin. You see, it's very easy. All right? Look at this. Here we go. Is this a thick cuticle? Yes, it's thick. It's a longitudinal section, and look how it's kind of folded. First of all, you're going to want to practice learning your morphological characteristics, and then you want to add on to that even tinier morphologic characteristics. Okay? So, this is a thick cuticle. This one we can't see. It's thin. But you can see at the lateral area, and I'll tell you how you can tell it's the lateral area later, that the cuticle is pulled out into some points, some ali. This is a thick cuticle. Now, I could sit here right now and tell you about the beautiful structures that we're seeing here, but I want you to focus only on the thing that we're talking about. Okay? All right, thick cuticle. Do you see a cuticle? No. All right? Thin. Here we go. Here are those lateral ali. They're pulled out. And you people now are trying to diagnose it by the genus. You're telling your friend, this is Enterobius. You're trying to look good in your friend's eyes. Good. Okay, anyway, the cuticle can be different colors if you do different stains. Here we go. Here are lateral ali. Look at they're 180 degrees apart. But look at these ali. You know, here's the lateral area. Look at how these ali have dropped down, evolutionary for some reason, but these are called sublateral ali. You don't have to remember this. I just want to show you the diverse morphological characteristics they can have. Sometimes in the anterior end, they have these little cuticular things that are either like teat-light, or these look like little, you can snap something on them like a screwdriver. Huh? Okay? And then here we go. Here's a cuticle. It's thick, and then it comes out to a point. And you will have to remember if this is a cross-section of the worm or the longitudinal section of the worm so you can get a feeling for just what this cuticle structure is. Sometimes they're small little bumps. Sometimes they kind of come out like ears. Sometimes they're different sizes, and they differ in the space between them. Now let's talk about external ridges. Some worms, this is a cross-section, people. How do I know? Well, a cross-section of a worm, I mean, it's going to be round to oval, right? We're going to cut it across this way. Nematodes, you know, are elongate and pointed on each end. Now, if you look at 30 cross-sections, and when you're describing a worm, try to get the one that is the most cross-section if you can. You'll want to look at a cross-section. And then, if you have evenly spaced little teats on every section, you've got to think to yourself, well, if they're sticking out this way, they're running the length of the worm. All right? And so these, when you describe them to your contributor, are called external, because they're on the outside, cuticular, because they're made of cuticle, longitudinal, because they're going longitudinal ridges. External longitudinal cuticular ridges. That's what you tell him. All right? That is to be contrasted with this worm. And you'll find out why that's important later. But this worm, this is a longitudinal section. This is a section this way. It's not a cross section. So if you cut something this way and you have evenly spaced bleeps, you know that those are rings going around the body of the worm. You see, that's different than the longitudinal ridges. You have annular cuticular rings. All right? And describe it that way. Sometimes the cuticle in the inside, this is lateral cord, by the way. This is muscle. We'll get to these later. But sometimes laterally at the area of the lateral cords, it bulges in right here. Okay? And this forms a ridge. And this is called an internal cuticular ridge. See, it's easy, huh, isn't it? And sometimes, look at this. This is cuticle right here. Look at this real thick cuticle. Here we go, a reddish cuticle, and it's kind of like, it's not pointed or whatever, but that is an internal cuticular ridge. Sometimes there are lateral ali, and some pieces of the worm have some like plaques on it, bosses, if you will. This is ganglionema. It's the only one that has that. And notice, when you see two sections of worm, nematode, next to one another, don't tell your contributor, well, I see two worms. You know, many people do that. These things coil usually, so it's usually one worm. But, you know, if you see a section in the arm, I mean the forelimb of an animal and the back limb of an animal, you can think maybe I have two worms. But if they're close like this, unless it's real long, but like this is one worm. Here, this is Gnathostoma. It has a cuticular bulb, which has little spines, but there's also spines on the body. I'm just showing you the myriad of different kinds of things. Is this a beautiful worm or what? Huh? Now, let's talk about lateral cords. Lateral cords are found laterally. That's very good. And these are they, right here. This is the lateral area. You say, how do you know it is? It's because there is a ventral nerve cord here. But anyway, there is, if you will, hypodermis. It's kind of like a mesenchymal layer that lies between the cuticle and the muscular layer in your nematodes. It bulges laterally to form sometimes immense lateral cords like this. These are big lateral cords. The lateral cords would also be described as being divided into sublaterals. There are two of them. You see? Here they go. Separates the muscle. Those are lateral cords. Now I'm going to show you all different types of lateral cords. Look at this one. Do you see how this is like the same height, if you will, as the muscle? So this is a low lateral cord. Look at this nice little larva. Here's our muscle. There's muscle there. And look at the pseudocoelom is taken up by lateral cords. That's the intestine. Huge lateral cords. And sometimes you really don't have anything that sticks out as a lateral cord, but you have accessory cords. That's the hypodermis coming out. And sometimes you have strange nematodes that don't have lateral cords at all. And those are your aphasmids, which is a group that we'll talk about later. You see nothing. You know, this is all muscle-like, and then you have this nice little structure as well. That's lateral cords. One of the most important morphological characteristics of worms is muscles. You know, and people go around saying, well, he has Platy-Myerian muscles or he has Celomirin muscles, and you, a novice, is saying, what in the hell are these people talking about? Y'all, it's very easy if you use my rule. Okay, this is Platy-Myerian muscles. Now remember, muscles run longitudinally in worms, all right? If the muscle is big and flat, it's platy. Well, what is a big flat muscle? This is one muscle cell right here. The muscle cell has a contractile, which is eosinophilic portion on H&E, and a cytoplasmic portion. This is one cell right here. This is contractile, that's a cytoplasmic. This is one. Platy-Myerian is also Merimyerian. That means there are only a few of them. If you have these big flat muscles, you can't have a lot of them. Now, this looks different. Do you see all these muscle cells? This is Celomirin. It's sticking to the inside like a column. All right? It too has cytoplasmic and contractile portions. And the contractile portions kind of come up the side of it, you see? Do you see that, now this is important, do you see that the demarcation between the red contractile portion and the cytoplasmic portion is not smooth? You see that? People, this is what I mean by smooth. You see that? It's smooth. This is somewhat smooth, but this is really smooth. This is smooth, much different than that. That's your Platy-Myerian. That's your Celomirin. Piece of cake. Look, here we go. Look, this is one muscle cell. Do you see how that's smooth? Do you see how this is not? Okay, now look, you've learned something. You've got your money's worth. Now, sometimes, here we go. What kind is this? This is Celomirin, and so is that. Do you see how that's not smooth? Sometimes lateral cards as well will have associated structures in it. Look at these kind of gland cells. But how do you can tell lateral cards from muscle? Well, you go, it's red and white, and then all of a sudden it's all white, and it's got red and white. And then there's little ale here as well. Okay, lateral ale. The cuticle at the mouth of the worm is sometimes different. This is a strongile mouth. You know, think of it like a hookworm, if you will. It isn't a hookworm, but you know, it's kind of like that kind of chitinized buccal cavity, you see? And sometimes, this is Physiloptera. It has that nice little piece of cuticle that comes out and forms that dome around the lips. And you can see those in your tissue sections. The mouth gives rise to an esophagus. This is a rhabdotoid esophagus. Many larvae and some groups of worms have rhabdotoid esophagi, or esophagi. Anyway, they have a body, they have a little isthmus, and they have a bulb. That's your rhabdoform esophagus. Look at this one. You have like a little V chitinized area. You have a muscular area. You have a glandular area. Just to show you that there are different types of esophagi, and you want to look at your tissue sections. The esophagus gives rise to the intestine, right here. This is the esophageal intestinal junction. And the esophagus, usually in most nematodes, will be triradiate. What is triradiate? That's triradiate. And the cuticle from the outside comes down into the inside and lines the esophagus. So you can tell this is in the area of the esophagus. Hopefully you people are now looking at structures that we've talked about cute little lateral ale, huh? Celomirin musculature. All right, intestines. Here are intestines. They kind of look like human intestines in some areas. What do you think this structure is? Esophagus, huh? And this is an intestine. How can you have that? Well, some have cecae that come anterior. Look at these strange lateral cords. Look at that thick cuticle. Look at the Celomirin muscles. Of course, this is all going through your head as I talk. Huh? Let's talk intestines. The muscles are one of the most important morphological characteristics we use, and the second is the intestine. Now, intestines are usually large or small. This is a large intestine, okay? This is a small. Sometimes intestines, you know, worms slurp up juice in the tissues, and sometimes they get blood. Sometimes they get the iron from the breakdown of blood cells. Well, that's the tiny little bitty esophagus. You see that? So that's small, that's large. Now, how can we tell that that's the intestine and it's not this? Well, let's face it. You're going to have one intestine in a nematode. You may have other things in the pseudocoelom. So when you see 6 structures and 5 look alike and one doesn't, the one that doesn't is the intestine. Does that make sense? This, you know, you have to call, although this doesn't have a lumen, so you would say that that is the intestine. This is a large intestine. This is small. Okay? Sometimes the intestines are kind of humanoid-like, if you will. They are made of columnar cells. They have, you know, nuclei at the base, but there's individual cells. Okay? And then you have this kind. This intestine is composed of 2 cells, 1 cell, 2 cell, which is multinucleated. So you would describe this intestine as being large and composed of few multinucleated cells. Let's talk boys and girls. All right? My rule of thumb, remember, we're looking for an intestine in this worm. All right, so we have 12 things that look like this. We have 2 things that look like that. We have 1 thing that looks like that. That's probably the intestine. Okay? Let's look over here. We have 6 things that are basophilic, 1 thing with a lumen. That's the intestine. Small intestine, large intestine. Anytime you see more than 1 genital tube, you're probably dealing with a female. Okay? I'll repeat that because I have stepped all over these wires. If there's more than 1 genital tube, it is a female. If there's 1, it's a male. You want to tell your contributor you have an adult female whatever. You know, it makes you look good. This is a female, that's a female. Now, conversely, if you only have 1 genital tube, it is a male. All right? 1 genital tube, an intestine. 1 genital tube, it's an intestine. We're not going to go into the different, you know, the ovary versus the testis and whatever right now, but intestine, okay, 1 genital tube. There are males. And you'll want to tell your contributor you have both adult female and male parasites because it's important for his case. You know, there might be eggs passing, there might be microfilaria in the blood or whatever. Sometimes the male, sections of the male will have chitinized structures, which are the spicules, which are analogous to, well, it's a holdfast organ used during copulation. And here we go. This is the tip-in of a male. So you can see that in tissue section. And while we're talking about sex, let's talk about eggs, ova, and sperm. Now, there are 2 things that make me nervous as a parasitologist, and my residents will tell you that. That's the use of the word ova. Okay, it's not your fault. People started this a long time ago. All right? If there is something that doesn't have a shell, all right, it is an ova. So really, those things with shells that you see in feces are not ova. All right? They're eggs. Now, the only time you people will probably see ova is inside the ovary of a female, or excuse me, the uterus of a female worm which has not been fertilized. That's where you'll see the ova, so you're not going to see very many people. One thing that you will find both in male and female worms is sperm. The males copulate with the females, and she keeps sperm in her, an area of her uterus called the seminal receptacle for long periods of time. So you can see both sperm in the males and the females. Sperm and nematodes are eosinophilic, circular to ovoid things, bodies with a dark nucleus. These are sperm. That's sperm. That's sperm, and that's sperm in a male. Notice how the females are often larger than the males. And here are little larvae. So, you know, it helps you say that it's a female. Females have eggs. You're not going to see them in males. And look, here we go, eggs. And use what you learned in vet school. Sometimes you will find that you can identify the egg within the female and therefore identify at least the group of the worm by just looking at the eggs. Here are some reddish structures that are have red globular yolk in it. What do you think they are? It's eggs. This is a female. Sometimes eggs are very small and contain a larva. Sometimes the females contain larvae themselves. And you're going to use all these characteristics when you go to identify them. Here we go. Many times people don't know the difference between Microphalariae, which is kind of a larval form, right? And the true kind of larvae that you see passed out in feces. Well, Microphalariae are usually, they're just a sack of little nuclei. You really see no esophagus or other kind of structures. Whereas if you look at these things, there's more to it than just a group of little nuclei. And that's how you can tell your larva from your Microphalariae. Sometimes worms will lay uninucleate eggs in tissues, and they will start to divide into morulas, blastula, and later they'll find larvae. And in some of your lungworm larvae, you can look at the tail, because some genera of lungworm larvae have specific types of little hooks and points on the tail, and you can use that. Sometimes the eggs will be colored. Sometimes they will have larvae. Sometimes they will have larvae, and they'll have nice little filaments at either end. And you'll use this to match up what you think it is. Now, let us go through the groups of nematodes. All right? This group is the atrachids, which are first cousins to strongyloides. Excuse me, they're kind of pinworm-like things. And I have this all in your handout, but there's lateral alae. The musculature looks a little Platy-Myerian, kind of hard to tell. But look, you can see there are larvae inside of this worm. Okay? Sometimes muscle is so inapparent you can't tell. This is rhabdius, which commonly occurs in snakes and in amphibian lungs, larvae within the female. And notice, it has pushed the muscle so much. These are the lateral cords. You can hardly tell anything about the muscles. All right? But here, these are ova. They're later going to make the larvae. And that's how you can tell rhabdius. Strongyloides. Almost all species of animals have strongyloides. It's not only man and horses and chimps and stuff. But you can only find females. Notice there is an intestine and 2 genital tracts. You only find females in tissue sections. Here are your females, 2 genital. And they lay uninucleate to 3-4 cell stage morula, and these start to embryonate, and you later have larvae in the tissue. So look for larvae as well. Rhabdotoid Esophagus. This is Halocephalobus, which was once called Micronema, but they changed the name only so they could ask that, I guess, on future board questions, because they asked Micronema long enough. But anyway, as you know, that these are parasites that are free-living, but can be found in humans and most notably in horses. And look how this esophagus kind of looks different than that, but it has a rhabdotoid-looking type of esophagus, and they're found all over the brain. The female has 1 genital tract, as opposed to what I told you before. It reflexes as the ovary right here, so if you get a longitudinal section, and they usually lay uninucleate eggs, and they're tiny little things, which the egg embryonates in tissue and forms larvae, which grows up into female, and you have large numbers of organisms. Here's your pinworms. Everything has pinworms, people. I mean, it's not only chimps. Cockroaches have 3 or 4 different types, if you would like to study that. But anyway, they have this rhabdotoid type of esophagus. This is a glycerin-prepared kind of whole mount, but this you can find in tissue section as well, so use it. The pinworms usually, I say usually, have lateral alae. The musculature is Platy-Myerian. Okay, look at this, how pretty. What do you think? Is this a male or a female? Male. Intestine, this thing has a lumen, and this is sperm. Remember, eosinophilic globules with a dark nucleus. Look, Platy-Myerian muscles, this is 1 muscle cell, and look at those nice lateral alae. That's a male anaerobius. Look at this. What do you think this is? This is a female. These are reproductive tracts. That's your intestine. Look at the demarcation between the cytoplasmic and the contractile portion. Here is a female with eggs. Look, these eggs are starting to look like what you learned in vet school. Now, this is a pinworm from a mouse, and notice how the shell, has ridges on it. See how thick it is? And then look out in the feces. Don't be afraid to look in the feces and match this egg with that worm, because sometimes you can make the diagnosis by looking for eggs and feces. This is Ascaridia. Notice it has a very small lateral ale here. It's a female because it has all these tracts. Now, other ascarids, there's many ascarids in animals. This is the ascarid, a type of ascarid Anisacus, but also Contracecum and Porosecum. They are parasites of marine mammals, and the lateral cards kind of come out, and they can look like a butterfly wing. And you can find that both in the larvae and the adult. This is Ascaridia. Notice the muscle is Celomirin. You don't have that flat thing, and it's a female. Okay? This is Balis ascaris. See, this is definitely not Platy-Myerian Musculature. The problem arises when you get ascarid larvae. It's very difficult to differentiate between ascarid larvae and spirurid larvae. The spirurids are another group of nematodes. The reason being is they have intestines that look somewhat alike, and they also have Celomirin musculature. But these are different presentations of ascarid larvae. Here's Balis ascaris. Look at those nice little lateral alae, and the worm is about 50 micrometers in diameter. Here's your spirurids. Great worm. This is Physiloptera. All right? Look at these lateral cards. Most of the spirurids have huge lateral cards. I mean, this is much different than your ascarid. And if you look inside the worm, you can find massive numbers of eggs, which you will later find that they're characterized as being small, oval, thick-shelled, and containing an embryo or a larva. Okay? So we call them embryonated eggs, not larvated eggs. A larva is outside. The embryo is inside the egg. It's just a tiny little thing. But anyway, the musculature is Celomirin, very thick cuticle, large lateral cards, and the intestine is not unlike the ascarid type that has columnar cells. It is often folded on itself and sometimes has a microvillar border. Look, there are the embryonated eggs, which we know are characteristic of a spirurid. Here's Drachia. Here it is. Isn't it pretty? Look at that. Celomirin musculature. It has lateral ale. What do you think this is? Esophagus. Thank you. Okay, here's the anterior end. It comes in. It doesn't have that rhabdotoid esophagus. Well, let's look at this. We get this section. All of a sudden, we see these larvae inside the uterus of the female. Well, remember I told you spirurids had thick-shelled, small eggs with embryos. This is a spirurid. This is different. I mean, this is Drachia. It's one of the exceptions. It has larvae within its uteri. It does not have the typical spirurid egg. Here is Somasaur, okay, Habronema, and this larva, look at this. You can even pick up the type of musculature. It's Celomirin musculature, and it has, you may not be able to see it, and I don't want to fool with the focus, but anyway, there's longitudinal external ridges on the larvae, which you can use to differentiate between that and an ascarid larva. This is Turgo Dermatitis, another spirurid. It has the normal types of eggs that you're used to seeing. Look at the musculature. It's Celomirin. It's the one with the sublateral ale. This is Spirocerca. Pretty muscles, huh? Celomirin, you can see the contractile portion and the cytoplasmic portion. The intestines are composed of cupoidal to columnar cells with a large microvillar border. And what sex is this? Female. And look at the lateral cards, how strange they are. They're sublaterals, but they're just not symmetrical as you've seen before. I put this section in here to remind me to tell y'all that, you know, when we study worms, when we study the morphology of known worms, we want to have a beautiful worm. If you don't have a good worm, you know, that allows us to study what it's supposed to be if we were in heaven, then when you get these nasty things that, you know, are degenerate and stuff like that, it's very difficult, you know, if you don't know your basic anatomy. So, you know, get the good cross sections and try to correlate that with the other dead worms. Now we have a tray change. And we continue. Tetrameres, it's a spirurid. You know, and you look at this thing and you go, look, I can't even make it out as a worm. Well, yeah, you're right. It's hard to make it out as a worm because it becomes such a hypertrophic sac of eggs. But these are all the eggs. It's hard to pick out the intestine and stuff, but if you study it enough, you can. But look, it's spirurid type eggs. It is thick shell, small, oval, contains an embryo. And look, it's the one with those little filaments on it. Okay, so you can use that as well. This is Trichospira. Most Trichospiras have very, very poorly developed musculature. But you can look inside and see those spirurid eggs, and you can tell it's a female by the number of genital tracts. Here's another section of a spirurid. Physiloptera, remember that anterior end? Y'all are all used to that, but you can see that in tissue section and use it. Gnathostoma, it's the only one with this big bulb and these spines. And here's a cross-section of it. Look, the Celomirin musculature and the anterior end, it has these lateral ali. This is one that you all know. It's ganglionema. And you go, well, look, I can't make out those muscles. And I say, well, you know, I have a pretty hard time too. But put it in the group. At least you can go to the group. Look at those eggs. Small, thick shell, and contains an embryo. Now, one of the easiest groups of worms to identify is the Phalarids, because there's only a couple that do not have a small intestine. All the other ones now that I've shown you have had medium to large intestine. Phalarids have small. Look, that's small, that's small. What do you think the sex is? Male, sex, female. Huh? Low lateral cards. These lateral cards are kind of low, but they both have Celomirin muscles. So you can differentiate between a spirurid and an ascarid quite easily just by looking at the intestine. You know, but when you describe it to your contributor, describe the whole thing because he'll think you're really smart. Here we go. What's the worm? Can you tell about, well, first of all, the cuticle is so inapparent, it's very thin. What can you tell about that muscle? Not very much. But what you can tell is, look at that intestine, very small. And it's a female, and they have microfilaria in it. Look at this one, Celomirin muscles, but who cares? It has a small intestine. Huh? These are 4 cuts of a uteri. Pretty worm, huh? Male or female? Female, female. Male, male, male. Okay? Huh? But look at the pretty Celomirin muscles. Low lateral cards. That was Dirofilaria immitis. Some Dirofilaria have these ridges on a cross-section, and they're called external longitudinal ridges. And this is very important as a taxonomic, in taxonomic use, and these are Celomirin muscles. This is Dirofilaria. Dirofilaria has lateral internal ridges. Many worms do as well, okay? But Dirofilaria seems to have really big ones, okay? Now, when you have a beautiful worm, or ones that we've seen in the past, let me go back a little bit. Let me show you these. You see, these are pretty worms. I mean, these were alive at the time of fixation. And look right in here. You can see these nice lateral internal ridges, and you really don't have any problems with diagnosing that. But then, when your contributor, I mean, you know, if your contributor knows anything, he's not going to send you the nice worms. I mean, he's going to make the diagnosis himself. But it's these type of worms that they're going to send you, and you're going to have to learn, excuse me, to use what you've learned and correlate it. Look at these lateral internal ridges. You see that? You see how the cuticle kind of goes in? Now, this cuticle looks a lot thicker than it did in the normal, the living worm, and that's because when worms die, the cuticle kind of swells up a wee bit. But the musculature is usually one of the last things to go, whereas the intestine is the first. But this is a Dirofilaria. But look at this case. I mean, this is a tough diagnosis, people. But, I mean, this is a uterus, by the way. Excuse me. And yeah, a uterus here. There was a uterus here, and this was the intestine. Can hardly tell. But look at those lateral internal ridges. Huh? And it had a smooth cuticle. So these are your Dirofilaria. Here's a nice gross section of Stephanurus, which is another filarial nematode. And here they are. Stephanurus is a little bit different than the other ones I know because you see this intestine is kind of big. You agree with me? I mean, it's not that tiny little thing. But they have, you know, this is a female. It has microfilaria. And Stephanurus has something that no other worm has that I know of, and that is eosinophilic disc in the uterus with the microfilaria. You have a graduate student, get them to find out what it is. It would be neat to know. All right? Eleophora, you know, and you make the diagnosis grossly. I mean, you know, gee, I mean, here's an elk. It has this stuff. I like to use this because you kind of see the growth, and then you see the worms there. Okay, and higher magnification. Pretty worm, huh? Now you see, it's going through your mind now. You have a small intestine. You have Celomirin muscles. It's a filarial nematode. You know, you can forget it. This is a male, a female, a female. You don't have to have the microfilaria anymore. Here, we have a lesion. I won't tell you where it is because you'll play the vet game. But let's look at this larva. What kind of musculature is this? Look at the demarcation between the red and the clear. It's flat. I mean, it's very smooth. This is Platy-Myerian musculature. Now, to make it easy, there are only really two groups that have Platy-Myerian musculature. It is the strongyles and the rhabdotoids, the anaerobius, the oxyurids. And I have that in your handout. So if you see some flat muscles, you know it's one of two things. And then you do away with one, and you have the group. There's only one problem. There's three different groups of strongyles. You have your true strongyles, hookworms. You have your trichostrangels, and you have your metastrongyles. And we'll find, for some reason, metastrongyles have Celomirin muscles, where the other two group have flat. I mean, if I was designing them, I'd have all flat muscle. But anyway, this is a characteristic intestine of a strongyle, and it is described as being large and composed of few multinucleated cells. All three groups have this. The strongyles, the true strongyles, usually have mouths like this, but look at this musculature, people. Absolutely pathognomonic of a strongyle. All right? Look at the intestine. It's large. You cannot find cell junctions. There are multiple nuclei. Large and composed of few nuclei. And look at this. Multiple nuclei, and it's composed of few cells. Look at that muscle, that pink, how smooth it is. Look at it. It's smooth. And some of them have lateral internal ridges. That's ancylostoma. Here's another one. Sometimes the strongyles incorporate blood or blood pigments, but look at that muscle. You should never miss the different types of muscle again. Hopefully. Large intestine composed of few multinucleated cells, Platy-Myerian muscle. Same thing here. Look at that that's one muscle cell right there. So these are your true strongyles. Now, let's go to trichostrangels. This is not a very good, this muscle is a little bit different, so let's not dwell on it. But the intestine is large. You don't see many cells in it, so it's large, composed of few multinucleated cells. But what most trichostrangels have are external longitudinal ridges. Look at them. They're sticking out like that, huh? And this is a cross-section, so it has to be going the length of the worm. Let's look at this one over here. Look, it's Platy-Myerian muscles, and look at those little longitudinal external ridges. These are your trichostrangels. Here we go again. Look at that platy. This is one muscle cell, Platy-Myerian muscles. Here's the intestine. Here are the little evenly spaced cuticular ridges, and here they are, evenly spaced. And then when you're getting into the lateral area, they kind of flange out. You can use these minor morphological characteristics. This is Ostertagia. I don't know if you can see it, but they have regularly spaced external ridges here. It's a trichostrangel, a very small worm. Look at this one. This is Nipostrongylus from a rat. It's a trichostrangel. It has these external cuticular ridges of unequal shapes and sizes. This is a lung with a consolidation here, and you look, and it's adult worms and larvae. Of course, they're lungworms. They're worms in the lung, right? Okay, what sex do you think that is? Female. And notice how there are larvae developing here. Here is the intestine. Look at that. Large and composed of few multinucleated cells. But it's the muscle that's different. It's Celomirin. Look at that. That's a large intestine. This is a large intestine. Okay? This is Celomirin Musculature. You see it's not smooth anymore? Right here, you see? And that, even though it has that, it is still a strongyle. What do you think these are? Sperm. Large intestine. And look at this. Here we go. Sperm, the ova, the same slide that I showed you. I liked it so much I used it twice. Now, the nematodes that I've just shown you are phasmids. I'm about to go through the aphasmids. Now, you're wondering, what is a phasmid? A phasmid is like a little pimple around the anus of the worms. Have I ever seen one? No. Will you ever see one? Probably not. But they break them up into groups. And the phasmid nematodes, we've gone through, have different morphological structures than the aphasmids. Now, you want to know what the aphasmids are? Look in your handout. Trichuris, Capillaria, Anatrichosoma, Trichosomoides. Okay? This group is different because it doesn't have lateral cards. And the other way that it's different is that the female has only one genital tube. But that shouldn't bother you in diagnosis. This is Trichuris in a dog. Remember, there's two parts of the body in Trichuris. There's the little small anterior end and the very thick posterior end. Now, Trichuris, not all aphasmids, have a structure that none of the other ones have, and that's a bacillary band. Right here, this basophilic structure. And usually it's in close approximation with the tissue itself, and some people think they use it to suck up things. I don't know. But another structure that all aphasmids have is a stichosome. And that is a body that surrounds the esophagus. Okay? The esophagus is surrounded by a bunch of donuts. 3 or 4 will have one nucleus. And that's the stichosome, and it's like a gland cell that communicates enzymes into the esophagus. Stichosomes are found in Trichuris, in Trichosomoides, in Capillaria. So you can use that. If you see a section with the stichosome, you can forget about the kind of muscles these things have. You know it's an aphasmid, and then go through your groups. All right? What kind of muscle? Well, it's Celomirin muscle. Do we care? Not really. Let's talk about lateral cards. Here we go around the worm. Do you see any lateral cards? No. Aphasmids have no lateral cards. All right, this is a female. Here is a male. I just show you this is the large posterior portion. This is another stichosome from Capillaria. Look, it's usually basophilic. The lumen of the esophagus is right there. And there are nuclei associated with the hypodermis, which are hypodermal bands. But it's not those pretty hypodermal cards that you've seen. And this is both sperm and ova within a female. And here are the eggs on the outside. So look around outside of your worm to help you diagnose them. Here's capillary again, but look at that stichosome. And we have aphasmid type eggs. You know, here are the eggs. Trichuris and Capillary have the same kind of type of egg. It doesn't have an operculum. It has a bipolar plugged egg. There's mucinous material in there. An operculum is a little flap, just like part of the shell that pops open. These are plugged, and the aphasmids can either have eggs or not within the eggs. Larvae are not within the eggs. This is Anatrichosoma in the skin of the hand of a monkey. Notice it has hypodermal bands. The female has one reproductive tract, but it has a lovely stichosome. Is that pretty or not? There's a stichosome. Look at the esophagus lumen right there. And here's an egg of Anatrichosoma that you can find from scrapings or sometimes passed in feces. And you can also find these in tissue sections themselves. And Anatrichosoma has an egg with a larva. What is this? Well, nobody misses this diagnosis because this is Trichinella larvae within muscle. And you know that's common. Trichinella is another aphasmid. Look, it has a stichosome. There's the lumen of the esophagus right there. That's the that's the stichosome, so you can tell it's an aphasmid. This is Dioxima, which is another strange nematode. Look at these muscles, how different they are from what we've talked about with our phasmids. Dioxymas have these kind of membranes are mesenteric kind of like strands that go from the muscle cells to the esophagus and intestine. There they are. And they have a very strange esophagus. And look at these muscles. See, they don't have that nice cytoplasmic portion. So those are the nematodes, and I've given you a succinct little chart in your handout so you can use these things. Now you can use these morphological characteristics that I've gone through. Now, we're going to go through the other groups of parasites. And I have, not as much to say about each group because each group is somewhat different than the nematodes. These are the Acanthocephalans. Now, most everybody gets the diagnosis when you have an anterior end embedded in the intestine that has hooks. You see these things right here? I mean, it's not often that you miss that diagnosis. However, when you have a section of the acanthocephalan in the intestine, sometimes people who don't know the morphological features will miss it. But acanthocephalans have something that nobody else has, and that's called Limniski. Remember, the acanthocephalans have a proboscis. This is moved out hydrostatically by muscular kind of things that evert or invert the things. And these are the Limniski right here. Look at these round little pipe-like muscles around it. And these, push it out or in. Look at these muscles, how strange they are. Aren't these strange-looking muscles? You could never call this a nematode. So when you get just one cross-section of this little booger, and it has these Limniski and these strange muscles, you're not going to diagnose it as a Physiloptera. Huh? And, it happens, people. But now you know different. Here's another acanthocephalan. Look, the muscles are different. They're in different layers. And the eggs are much different than what we've seen in nematodes as well. Mites. I don't get excited about mites. This is pneumonisis. All you people make this diagnosis by looking at the mite pigment in the lung. But this is a nice little picture because it shows the chelicera and the palps of the mite itself. You have this red globular material inside this worm. What do you think? This mite. This is an egg. This is a female. Here we go. Look at these areas are highly chitinized. But if you go on high magnification in areas like this, you will find that the muscle is striated. Remember, I never used striated muscles so far. This is the first group that we've seen that will have striated muscles. Here we go, and you can pick it out right in here. Those are the little H bands, Z bands, I forget my numbers. And some mites will have little spines on their dorsal area. And that, of course, is Sarcoptes. And most mites have 2 pair of legs in the posterior part and 2 in the anterior part. But Demodex, which has a long abdomen, will have the legs starting from the origin of the legs in the same area on both sides. You see there's no split. So you can tell Demodex from all of the other types of mites. Bots and maggots, which are larval flies. I just love this. I call it Dracula. Anyway, they have spines on the cuticle, and you certainly can pick that up in your histologic section. And let's say everything is degenerated and you see some spines, you could possibly make the diagnosis. Look, this has striated muscle as well. There is a breathing apparatus that the maggots and bots use, and that's called a spiricular plate. And kind of think of it as an air conditioning vent cover, and that's to the outside, and it sucks in air. And these give rise to a cuticularized tree of tubes, which brings the air to the inside. Here's a section of one of those spiricular plates. Look how it's chitinized. And then it goes down into these small tubes within the inside of the mite itself, the bot itself. Another phylum of worms are Penta Stones. These are once thought to be mites because their first larval stage is identical to a first larval stage of a mite, but we now know that it is another phylum to itself. Pentastomats have hooks on the anterior end. They have intestines. And they have eosinophilic glands, which follow the intestines probably for the first half of it. Now, here are the eosinophilic glands. I just point these out because no other animal has these things. They're called pentastomats because they usually have 2 pair of hooks and a mouth, which Pentastomids means 5 mouths. They thought that these were kind of mouths as well. Now we know they're hooks. And you can pick up these hooks in section as well. And you usually have nymphal stages within mammals. Now, Pentastomids have a structure that no other parasite has, and that's sclerotized openings. When you look at the cuticle or the body wall of a pentastomid nymph, you can't see these things. Look, you see a little eosinophilic thing. Use a Movat, and you can see that the opening to the cuticle turns black. Okay? No other animal, no other parasite has this structure. So you say, is that useful? You better believe it's useful. You have this thing in the liver of an animal, you do a Movat on it, and look, you start seeing the sclerotized openings. Huh? You see that right there? And so you say, well, I could have made that diagnosis anyway. Yeah, you can, but then what if you get this big nodule, and you see something in there, and it's just eosinophilic cuticle, and you call it an ascarid that has migrated into the liver of the animal? Well, you get a Movat, and then you start seeing these brown little circular things, which are the sclerotized opening of the cuticle, and you know that this was a pentastomid nymph. Trematodes. Trematodes are very difficult to find the genus, and definitely species, in tissue sections. The trematodes have vitellaria, which are yolk-producing cells, usually on the periphery. They have intestines, they have testes, they have ovaries, and they have suckers, and then they have uteri that are just filled with eggs. They have a solid parenchyma, which is like a sponge, so you don't see any of that pseudocoelom or coelom like we've seen in the others. And this is a cute little thing. I mean, you know, here's the anterior sucker and the ventral sucker, and he's holding on, and notice there's an intestine here. But look for eggs. I mean, trematode eggs are described as being small and brownish. Here are 2 cechi, or 2 intestines, in the vitellaria on the periphery. This is to get rid of waste. Here we go. Here's vitellaria on the periphery. And here, this is what they look like. I mean, you know, it's not a hard diagnosis to make. But now, if you have this thing migrating in a sheep, you'd probably know what it is. You'd play the vet game and you'd say it's so-and-so. But if you have an unknown animal that has an unknown one of these things, you really can't go more than trematode. Here we go, a schistosome and a blood vessel. And, you know, most people think that all schistosome have either terminal or lateral spines like Schistosoma mansoni here. But in fact, there's so many other types of schistosomes in the world. I mean, this is a turtle schistosome, if you will. Huh? You see these brown eggs all in the parenchyma of this turtle? And here, this is the schistosome that occurs in the United States, and that's Heterobilharzia. I mean, you know, it is not schistosoma. There are other genera of these things as well. Cestodes, the adults, you really can't do anything about them on tissue section. You can call them an adult cestode. Sometimes they have hooks or arms on their proboscis. These are suckers, not eyes. And here we go. They have parenchyma as well, just like the trematodes. But cestodes have something that no one else has. And, you know, I don't know who devised all these things, but I thank them because I use them all the time. And that's calcareous corpuscles. That are little football-shaped kind of mineral concretions in the parenchyma of the organism. And you can pick that up, and that's what allows you to call it a cestode vice a trematode, although cestodes have no digestive tract and trematodes do. But they usually are segmented. Look at this. Here are the segments, the adults and the intestine. And that's how you can tell it from a trematode. Now, there are different types of larval forms that can form in animals. And this is a cysticercoid, which is in a beetle. And it's very difficult to find in a mouse intestine. But this is one. Here's suckers. Here's a little rostellum, and it's in the villus itself. And cysticercoids are very, very rare, as opposed to cysticerci, which all of you people know, you never miss the diagnosis. It is a balloon which has one scolex, or one neck, with a rostellum. And here is the bladder. Here is the neck. And those black things you will find later to be calcareous corpuscles. Remember, the thing that nobody else has. All right, there they are. And sometimes you have hooks on the rostellum. It is armed, and sometimes you don't. Here is a Sonuris, which is just like a Cysticercus, except it has more than one scolex. And here is the bladder, and here are the multiple scolices. And this is a hydatid cyst with daughter cyst, okay, and protoscolices, and very nice laminated thick wall in one of the types. And sometimes, you know, it's very old. This is a pig lung, and you find this laminated structure in the lung, and you diagnose it as an old cyst wall of entamoeba. Where have I been? Echinococcus. All right. This is the multilocular type. It kind of goes through like a cancer. It doesn't have that big laminated wall like you've seen before. But certainly it has aggregates of protoscolices like we saw in the unilocular. And then you have the intermediate form, which is Echinococcus vogeli, sometimes Echinococcus oligarthus. And you have somewhat of a laminated outside, but then you have little septa, like you would have seen in the multilocular cyst. Okay? And this is, you know, only 5 years ago recognized. Another form of larval tapeworm is a Sporganum. Notice the sarcocystis. Anyway, here's a Sporganum, and it's just solid. It does not have a scolex, and it's found in connective tissue and muscles of many types of animals. Here's a nice little mouse, and notice these little kind of cystic things, and I tease them out. Here's a cyst, and here's one out. And we find that it has a bladder and a scolex, and the scolex has a rostellum and suckers, and that's a Strobilocercus. Okay? All different kinds of larval cestodes. It's a varied group. It's much different than just one Cysticercus. A group that I'm interested in is larval cestodes that seem to proliferate. This is a pearly nodule in the omentum of a monkey, and notice it looks kind of like a Sporganum. But then, this is a cat from Falls Church, Virginia. Here's the stomach. This is the epithelium. And notice, look, it's a Sporganum-looking thing here in this muscle, but then we go over here, and it looks like a cancerous kind of Sporganum. This is the dog, same kind of thing. Epithelium, and look, nice Sporganum-looking things. However, they have all these vacuoles and things. And this is much different than we've seen in either one. I have no idea what these things are. It's a good study for a graduate student, if you have one, to collect all the cases of these proliferating larval cestodes to determine. Okay, so that's morphology of worms. Now, I'd like to leave you with two cases. I always like to show you interesting things. This is the intestine of a radiated tortoise from my dear friend Elliot Jacobson. And notice, I would suggest that these things are normal epithelial cell nuclei. But notice how they start to get kind of bigger. They don't have as much chromatin. And then they start to have things within it. Okay, what do you think those are? Since I'm talking, I don't think you'll call it bacteria. Anyway, here we go, and then we have this larger thing that has all these dots. Huh? Look at how many. Now that your eyes are attuned to it, you can see they seem to be multiply infected. These are 1 micrometer sections, by the way. This is not a normal 5 section. And then you can see some things that look like they have a shell and eosinophilic globular material. So what do you think it is? A coccidian, right. We did EM on it. Look, here's the host cell nucleus, and you see all these zooites from a schizont. Hey, isn't this pretty? You see, and there's different schizonts. They're at different stages of development. This is mature, this is not. Here we go. Look at how they're budding from a residuum. Pretty little things, aren't they? Look at that. And they have all the structures, of course, of apicomplexa, right there. And this is the host cell nucleus. I had never seen a coccidian in the nucleus. I mean, you know, usually they're in the cytoplasm of the epithelial cell. Look at this. What do you think this is? Well, this turns out to be a macrogametocyte. these areas are those eosinophilic globules which will form the oocyst. So we had both the female, and this is the male, and unfortunately you really can't pick it up, but there are little flagellae in here, you know, for the microgametes to have, and this is a developing microgametocyte. So there's only been 12 cases of intranuclear coccidial infection. So this will soon be published. Another case that will soon be published, which I'm very interested in. This is a nodule from a dog from California. I was told that this is the dog in Dances with Wolves, the dancer, if you will. He presented with this nodule from his eye, and there it is in higher magnification. You know, when you see nematodes, you know, I mean, these things kind of look like worms. You know, you have like a little clear area and a kind of a whitish area. The whitish area usually is the worm, the nematode that's coiled around. You see that? And the clear area is kind of like the reaction, the fibrosis or whatever, to the worm itself. And then we do a section, huh? And we see, you know, how many worms do you think this is? Well, it could be one, it could be two or three. I mean, this is probably one, this is another one. But we have this worm. Now remember, this is a dog from the United States. And now, as we're working up that case, we get another case. Dog being from the California area. Eye, this was an incidental finding, the outside of an eye. And look, we have worms, so we get excited. Some worms excite us more than others. All right? Determine. Okay, so that's morphology of worms. Now, I'd like to leave you with two cases. I always like to show you interesting things. This is the intestine of a radiated tortoise from my dear friend Elliot Jacobson. And notice, I would suggest that these things are normal epithelial cell nuclei. But notice how they start to get kind of bigger. They don't have as much chromatin. And then they start to have things within it. Okay, what do you think those are? Since I'm talking, I don't think you'll call it bacteria. Anyway, here we go, and then we have this larger thing that has all these dots. Huh? Look at how many. Now that your eyes are attuned to it, you can see they seem to be multiply infected. These are 1 micrometer sections, by the way. This is not a normal 5 section. And then you can see some things that look like they have a shell and eosinophilic globular material. So what do you think it is? A coccidian, right. We did EM on it. Look, here's the host cell nucleus, and you see all these zooites from a schizont. Hey, isn't this pretty? You see, and there's different schizonts. They're at different stages of development. This is mature, this is not. Here we go. Look at how they're budding from a residuum. Pretty little things, aren't they? Look at that. And they have all the structures, of course, of apicomplexa, right there. And this is the host cell nucleus. I had never seen a coccidian in the nucleus. I mean, you know, usually they're in the cytoplasm of the epithelial cell. Look at this. What do you think this is? Well, this turns out to be a macrogametocyte. These areas are those eosinophilic globules which will form the oocyst. So we had both the female, and this is the male, and unfortunately you really can't pick it up, but there are little flagellae in here, you know, for the microgametes to have, and this is a developing microgametocyte. So there's only been 12 cases of intranuclear coccidial infection. So this will soon be published. Another case that will soon be published, which I'm very interested in. This is a nodule from a dog from California. I was told that this is the dog in Dances with Wolves, the dancer, if you will. He presented with this nodule from his eye, and there it is in higher magnification. You know, when you see nematodes, you know, I mean, these things kind of look like worms. You know, you have like a little clear area and a kind of a whitish area. The whitish area usually is the worm, the nematode that's coiled around. You see that? And the clear area is kind of like the reaction, the fibrosis or whatever, to the worm itself. And then we do a section. Huh? And we see, you know, how many worms do you think this is? Well, it could be one, it could be two or three. I mean, this is probably one, this is another one. But we have this worm. Now remember, this is a dog from the United States. And now, as we're working up that case, we get another case. Dog being from the California area. This was an incidental finding, the outside of an eye. And look, we have worms, so we get excited. Some worms excite us more than others. All right? So, now, let's look at the morphological characteristics that I've taught you. All right, what's the intestine like? Small. You see, y'all have learned something. You have got your money's worth, and you didn't think it was worth it. Anyway, small intestine. So what group is it? It's a filarial nematode. Well, you think it's a filarial nematode. But let's look at what sex. Female, huh? And what does the female have in its uteri? Microfilariae. That's what it is. I mean, it goes so the whole package is complete. It's a filarial nematode. Now, you people who would like to play the vet game, huh? What would you say? Well, you would be a fool not to say Dirofilaria. You know, ectopic Dirofilaria immitis in a dog. I mean, I would buy it for a little while. To continue, here's another section, okay? This is the intestine here. This is the intestine. It's a female. These are microfilaria. Let's talk about the muscle, just because we're here. Well, the muscle is here. It's Celomirin, but what's happening, people? Those lateral cords, huh? These are the lateral cords. Do you see how they're kind of taking over the muscles? I didn't show you any cases like that, did I? So it seems like the worms are sitting around, and the muscle is becoming hypotrophic, and it's kind of dying, and the hypodermis is taking over. Will you buy that? Okay. Here we go. Now we're going to do a longitudinal section. Think, longitudinal section. And we have evenly spaced bumps. Now, are those external longitudinal ridges? No. Those are what? They're annular rings. They're going around the worm. There is only one worm with annular rings. Okay? And that is Onchocerca. So, what's Onchocerca doing in two dog eyes? Well, let me show you some more sections. You know, you can go into your tissue when it's fixed, and you can tease out some pieces. You know, that means you take a probe and you take it out. And we look at it in glycerin, and not only in longitudinally do we see these evenly spaced ridges, but we also see something else underneath, part of the cuticle. You know, there's another layer of cuticle here, and it's doing something strange. Okay, let me just back up right here on the H and E and show you here. Look, you can see them here. Huh? And these things have a different kind of relationship to these bumps. There is one under each bump, each annular ring, and then there's one in between them. These are called striae. And you would say there is one stria in between each rings. Now, this was worked out some years ago by Dr. Paul Beaver at Tulane University. But look, here it is again. This is the other worm. You see how there's one stripe between the annulations. Look at this. Beautiful. Look. Here's one. There's one under there, one under there. Thing. Now you say, well, you know, you're kind of getting picky. Well, you know, you need to be picky because the species of the Onchocerca is determined by the number of striae between the annular rings. All right? So a year and a half ago, there was one case from a dog from California. Now we have two more cases, and we have just received the fourth case from a valley south of Los Angeles. They must know that I'm in San Diego, you know. But this has great importance in not only animal research, but in human research. Onchocerciasis is one of the top 5 infectious diseases by the WHO for people, okay? You know, because of the morbidity, mortality associated with the infection, and that we cannot control it. We are always searching for ways to come up with better drugs. We have been hampered by this for years because we did not have a suitable laboratory host. We've tried to infect non-human primates, didn't work well. One was infected. Now we know that cattle and horses are infected, but cattle and horses don't make the best laboratory animals we have seen. You know, they eat a lot, they go to the bathroom a lot. Well, I don't know, y'all do laboratory animals. But anyway, this has lots of repercussions because if we can find out what species this is and try to infect dogs and have it take, it is a wonderful animal model, and it means big bucks to the person who does it. So because we have 4 cases, we have an epizootic, you know, in dog eyes. And what's strange about it is the problem of onchocerciasis in humans is also an ocular one, but it is due to the microfilariae, which is found, of course, in the, you know, in the cornea, causes blindness. And now all the cases that we have, there are only 4, the adult worm is always associated with the eye. Now, we have never had an autopsy on one of these dogs to know if there are nodules, there are worms elsewhere. Now, there's only one more thing that we would like to know happens in these cases, is that, of course, there is male and females, because this female is filled with sperm people, and they also have microfilariae inside the uterus. We want to know, does the microfilaria go out into the collagen, into the skin of the dogs, so we can try to use topical anthelminthics to cure the disease? And by happen, it does. This is a microfilaria. So now, if we can decide which species it is, we have the answer. And I feel that it's Onchocerca linalis from cattle. We have tried to infect dogs numerous times with Onchocerca volvulus to make a laboratory animal, an animal model, and we have always failed. But we have never taken linalis and tried it in dogs. So my friends who have black fly colonies, who have access to microfilaria of linalis in cattle, are getting the linalis microfilaria, feeding them to the black flies, keeping the black flies alive for a couple of weeks, seeing if they have infective larvae. If they do, injecting it into dogs and waiting to see how long it takes, or if it does take, to get the worm in the dog eye. And when it does, unfortunately,